Hey everyone! In these videos, we are going to review major topics in AP Biology, but we're going to do it from a different point of view. For example, in this first video, you're going to be reviewing carbohydrates, but rather than reviewing carbohydrates just in Unit 1, we're going to go through and look at how carbohydrates apply to all the units. So we have 6 major units that we did in AP Biology. And so for each of these, we're going to be looking at how carbohydrates spiral through each of these units. We're going to be looking at first at the structure and function of carbohydrates in Unit 1. Then in Unit 2, we're going to talk about the role of carbohydrates in cell membranes. We're also going to talk about, in energetics, how breaking down and building of carbohydrates occurs. In Unit 4, we're going to talk about the role of carbohydrates in certain genetic examples. Then in Unit 5, we're going to be looking at a specific example of an operon, which involves the breakdown of the carbohydrate lactose. And then for Unit 6, evolution, we're going to be looking at carbohydrates that are in Hardy-Weinberg equilibrium. So starting with Unit 1, macromolecules, we started the year by looking at the structure and function of carbohydrates. So we're going to focus on first the chemical structure of carbohydrates. So remember that they're made of carbon, hydrogen, and oxygen in a 1-to-1 ratio. And keep in mind that this oxygen right here is very electronegative, which means that when it's paired with the hydrogens, it's going to pool those electrons a little bit more and create those slight polar ends. The bonds of carbohydrates are glycosidic bonds, and the monomers are called monosaccharides. Sometimes you can have disaccharides, which means 2 carbohydrates placed together, and then a polysaccharide is going to be 3 or more. In all of these units, we stress the importance of understanding of how structure affects function. And a good example in carbohydrates that comes up many times on the AP exam is the difference between cellulose, starch, and glycogen. Now, all 3 of these molecules are polymers that are made up of glucose. So they are long chains of the glucose monosaccharide. But because their structure is slightly different, they're going to function very differently. For example, with cellulose, it's made up of a form of glucose called beta glucose, whereas starch and glycogen are both made up of alpha glucoses. Now, in addition to the structural difference of the glucose molecule, that is the direction in which the hydroxyl group is facing at carbon number 1, cellulose, starch, and glycogen are also different in their branching. Now, cellulose is completely unbranched, and this makes up the plant cell walls. It's a very tough molecule. It's not digestible by people because we don't have the enzymes to break down cellulose. Whereas starch and glycogen, which are made of alpha glucoses, they are branched. In the case of starch, it's slightly branched. In the case of glycogen, it's highly branched. And starch is used as plant energy, whereas glycogen is in muscles and in the liver and is animal energy. And this difference in their structure, both in whether or not they contain the hydroxyl group or which direction the hydroxyl group is facing, and whether or not they're branched, is going to make a large difference in whether or not they function as a food source, such as starch and glycogen, or if they are more structural like cellulose. Now, in the case of the bonds that are formed in the carbohydrates, that glycosidic bond is either broken or added, and it's important that you remember that whenever these molecules are being made or they're being separated, that it is the involvement of water. So either dehydration synthesis or hydrolysis. So here are 2 generic monomers for carbohydrates. To keep it simple, I did not draw all the hydrogens and oxygens, but I do want to call your attention to these 2. Because from these 2 monomers, when we are building a polymer up, we're going to need to remove water from these 2. Now, what's going to be left behind is one of these oxygen groups. So remember that water is H2O. Here's H number 1, here's H number 2, and then there is the oxygen group. So what's left behind is that oxygen right there, and that oxygen is going to be shared by these two carbons, and that is what forms the glycosidic bond. And so that glycosidic bond was formed from the removal of molecules both from this subunit and this subunit, producing water as a byproduct. The opposite would be true if we were going to break this bond. We would split the water to restore the original OH on this side and OH on this side. Now in Unit 2, carbohydrates played an important role for membranes. So carbohydrates are part of what is known as the fluid mosaic model. Carbohydrate chains are on the surface of the membranes and act in identification, more specifically with those blood types that we're going to talk about a little bit later. Also, carbohydrates act as part of the extracellular attachments. If they're attached to proteins, they are known as carbohydrate proteins, or what is known as a glycoprotein. And if the carbohydrate is attached to a lipid, it is known as a glycolipid. Due to their large size and polarity, carbohydrates such as glucose cannot freely pass the membrane and will need special channels to get through. Remember that if they're going down their concentration gradient and they just need the special channel to be able to pass through the membrane, this is known as facilitated diffusion. If the glucose or carbohydrate is going up against its concentration gradient, then it would be an example of active transport. But in this case, if we're talking simply about carbohydrates moving down their concentration gradient with the help of a special channel, it is simply known as facilitated diffusion. Carbohydrates also make up an important part of cell walls, as we had mentioned in the Unit 1. So we know that cellulose is in plants. There's also another carbohydrate that is called chitin, which makes up the cell walls in fungi. Now, chitin is also different because it contains nitrogen. There's also a protein carbohydrate blend called a peptidoglycan, and that's going to make up the cell walls in certain bacteria. Now, Unit 3 was very big for carbohydrates. In Unit 3, we talked about the breakdown of carbohydrates, which is the cellular respiration piece. We also talked about building up carbohydrates, which is photosynthesis. So in the breakdown of carbohydrates, which is known as cellular respiration, we start with glycolysis. Now, glycolysis is overall an exergonic process. This means that it has a negative delta G. It's catabolic. It's going to break down glucose, which is the starting molecule. There is an investment of 2 ATP, and the ending molecule of pyruvate is a 3-carbon molecule. During this process, there's also going to be the reduction of NAD plus down to NADH. And we're going to produce 4 ATP by a process called substrate level phosphorylation. Overall, the process of glycolysis is anaerobic. It does not require oxygen, and it's regulated by enzymes, 10 enzymes in the cytoplasm. Now, because this is an anaerobic process, all organisms use the same exact 10 enzymes, and it occurs in the cytoplasm. Those are pieces of evidence that glycolysis was the process performed by Luca, the last universal common ancestor. In the presence of oxygen, that pyruvate molecule will undergo further oxidation and it will go through the Krebs cycle as long as a mitochondria is present. So overall, the process of oxidation of pyruvate is also exergonic, meaning it's got a negative delta G, and it's going to start with that pyruvate molecule. That pyruvate molecule is going to get oxidized, and it's going to get a coenzyme added to it. So what we're going to end up with a molecule of acetyl-CoA. Now, acetyl-CoA is going to enter the Krebs cycle in the matrix of the mitochondria, and it's going to join with a 4-carbon molecule called oxaloacetate. So what we end up is a starting molecule, a 6-carbon molecule called citric acid. And so sometimes this is referred to as the citric acid cycle. During the citric acid cycle, or your Krebs cycle, we're going to reduce even more NAD plus and FAD plus. There's also going to be the substrate level phosphorylation that occurs. And so we get 2 more additional ATP formed from 1 molecule of glucose as the products go through the Krebs cycle. It is once again regulated by enzymes and occurs in the matrix of the mitochondria. And this occurs in every eukaryotic cell. And it's worth noting, this does include plants. Plants will go through cellular respiration as well. Now, the NAD plus and FAD plus that had been reduced in glycolysis and the oxidation of pyruvate and the Krebs cycle are then going to be donated to an electron transport chain in the inner membrane of the mitochondria. That electron transport chain is going to use the electrons that are taken from NADH and FADH2 and use it to create a hydrogen ion concentration gradient. Now, thanks to this hydrogen ion concentration gradient, we're now going to produce ATP as ATP flows down its concentration gradient. This process is known as oxidative phosphorylation, and it occurs in the mitochondria, thanks again to that electron transport chain creating the gradient difference. Overall, this process is endergonic, meaning that there's an input of energy to make ATP, and that energy is going to come from what's known as the proton motive force. In other words, as the protons are moving from a high concentration to a low concentration, ATP synthase is able to use the movement of those protons to phosphorylate ADP into ATP. Now, the second part of Unit 3 was making carbohydrates, and they're made through a process called photosynthesis. Overall, this process is endergonic, meaning energy input is going to be needed. Now, this energy is going to start by coming from sunlight, and it's going to be the light-dependent reactions that capture the sunlight, or capture the energy from the sunlight. They use sunlight to oxidize water, producing oxygen as a product. So we're going to strip the electrons from oxygen. O2 is released as a product, and this is going to occur at photosystem 2. There is also an electron transport chain that creates that hydrogen ion concentration gradient to produce ATP using proton motive force, just like we saw in oxidative phosphorylation. Although with this one, because it's going on during photosynthesis, this is referred to as photophosphorylation. Now, this is occurring in the thylakoid membrane of the chloroplast, and our final electron acceptor, which is NADP+, is reduced down to NADPH, and ATP and that NADPH are going to then power the dark reactions. Now, the dark reactions are also known as the Calvin cycle, or your light-independent reactions. They occur in the stroma, and there are 3 main phases. Phase 1 is known as the carbon fixation phase. This is where CO2 is taken out of the atmosphere and attached to an organic molecule. The second phase is known as the reduction phase. In this phase, we are going to need the electrons from NADPH and the energy of ATP to continue rearranging the molecules. The reduction phase is essentially the reverse of glycolysis. Now, ultimately, we are going to eventually produce glucose at the end of the reduction phase. But we also need to make sure we are ready for the next turn of the Calvin cycle, so we're going to go through the regeneration of the RUBP phase so that the Calvin cycle is ready to turn again with the next carbon dioxide molecule. It's going to eventually take 6 turns of the Calvin cycle to produce a glucose molecule. Now, in Unit 4, cell division, we are going to look at the specific example of carbohydrate chains on the surface of red blood cells, and remember that they are inherited in a Mendelian pattern of inheritance. So your blood typing for A, B, AB, and O blood typing are indicating a particular carbohydrate carbohydrate chain that's on the presence that's on the surface of red blood cells. And these red blood cells, or these carbohydrate chains, are inherited in Mendelian patterns, meaning that you can use Punnett squares, dihybrid crosses, or even pedigrees to be able to predict chances of offspring having particular phenotypes. Now, with the AB blood typing, this is also an example of codominance. And with codominance, what happens is if a person inherits the genes for both the A and the B type chain, they will have both chains in their red blood cells. So they will have both A chains and B chains. And so the heterozygote condition for AB blood typing is different. Now, the other interesting thing about carbohydrates in terms of genetics is that human blood type is in Hardy-Weinberg equilibrium. And we'll talk a little bit more about this on Unit 6. In Unit 5, which was molecular genetics, we used the specific example of the breakdown of lactose in prokaryotes, which is known as the Lac Operon. Now remember that operons are clusters of genes that produce one mRNA molecule, and from that one mRNA molecule there'll be several protein chains encoded. In this case, it's going to be the proteins that come together and form the enzyme to break down lactose, which is called lactase. Remember that you can tell something is an enzyme by its ending of ASE. Now, the Lac Operon is what's an example of an inducible operon, meaning that the operon is normally turned off, so the production of the mRNA is not activated normally. But under the right conditions, the Lac Operon, to produce the proteins needed to make the enzyme lactase, can be turned on if lactose is present and glucose is absent. Operons, keep in mind, sound very similar to transcription factors that we talked about later, but operons are specific to prokaryotic cells. As I mentioned, human blood type, which is based on a carbohydrate chain on the red blood cell, is in fact in Hardy-Weinberg equilibrium. So when we look at the evolution of species or evolution of populations, we have to remember that with Hardy-Weinberg, there are certain conditions that must be met. In the case of carbohydrates, or specifically the carbohydrate chains on the red blood cells, this is an example of a gene that is in Hardy-Weinberg equilibrium. So let's take a look at the genes and let's take a look at the conditions and how this condition of human blood typing, the A, B, and O blood typing, are in equilibrium for the human population. Number one, remember that for Hardy-Weinberg equilibrium, there can be no mutations. And this is true. You don't read in the news reports or see posted on Facebook that a new blood type has been discovered. So no mutations is something that these red blood cells, or the carbohydrates on those red blood cells, is following. The next is no migration. So in our case for humans, that would mean other humans from a different planet are coming in and bringing in different ratios of these blood types. Now, depending on the population you are looking at, if you're looking at, let's say, an isolated island somewhere, then you would probably say, oh, this is violating the no migration condition of Hardy-Weinberg. But in this case, we're looking at humans as a whole, like a whole population. And so since we don't have anyone coming into the planet, nor is anyone leaving the planet and never coming back, we are still okay with that condition. No natural selection. So there is no blood type that does not make it easier or harder for people to survive. Now, I've heard people say that certain blood types attract mosquitoes more, but I don't know that there's enough evidence to support that in the case of that a predator is specifically wiping out a particular blood type. And the blood type does not affect your reproductive abilities either. And speaking of reproductive abilities, blood typing is not something that is selected for in mating. So this blood typing is going to follow the random mating rule. As of this point, I don't think that this is a question that's asked on Tinder. But if it were, then we're looking at sexual selection that's affecting whether or not your ability to pass on those traits is compromised or not. And then the last condition is large population. And I can guarantee you that 7.6 billion people on this planet is considered a large population. At least that was what it was when last I checked. All right, so that does it for your review on carbohydrates. We're going to have other videos where we go through and look at major themes in AP biology, and again, spiraling those themes throughout the units that we have covered. Thanks y'all so much for watching. Bye.